Hey there. So I wanted to go over lab reports two's rubric, right? So that way you guys know exactly what I'm expecting, and it's the same rubric roughly with lab report one. I just put a little bit more text box, a little bit more side notes of what I'm expecting, so that way it's really clear. Majority of you guys did great for lab report one. I just put a lot of commentary about like what I was really expecting. So what I would consider what you guys have turned in is a really good rough draft for lab report one. And then had you go over it again and took those commentaries to heart and really fixed up those little issues or added in the additional information that I was asking for, it would have been like a perfect lab report. So that's what I'm kind of expecting for lab report two. Take in the feedback that I've given you, and if you haven't got that feedback, I'm really sorry. I'm going through a little bit of stuff over here. But just watch the feedback video. That's why I went ahead and posted that one. So that way you know what I'm really expecting, or watch this next one because this one will go over what I'm expecting for this next lab report, right? So let me go ahead and share my screen. All right, and here it is. And as you can see, it's roughly the exact same, lab report. It has the same kind of breakdowns with an abstract, a cover page, an introduction, methods, results discussion, references, and overall. You have the option still to go to the Writing Center. I saw that they are taking it virtually, but if you are going to do it via a Writing Center, you cannot turn it in the day of. So I need to double check, but I think that your lab report 2 is due April 30th. Don't quote me on that, check the syllabus, but I'm pretty sure it is due April 30th. So if you were to have sent it via email or virtually to the Writing Center on April 30th, it won't get done in time. You need to send it, have it done by that Monday or something like that. Give them 3 to 4, maybe even more, maybe a week ahead, a good amount of days to really look it over, because I know they get really swarmed with a lot of lab reports at this kind of period of the semester, right? So you want to give them plenty of time to get back to you. Just fair warning. And that would give you those 5 extra points. Turning it in early will give you those extra 5 points. Those are 10 extra points that really came in handy for lab report 1 with you guys. So I'll go ahead and leave them again for lab report 2. All right, but as you can see that you would start with a cover page. You want to put in your name, lab date, and time. One thing I will I need it to be shown is that if you did it individually, it should just have your name, and then even right there, I would even put on there like done individually by Sarah Bustamante or something like that. But if you did it as a group, you can put like done as a group, group 2, which is made up of the following, Sarah Bustamante, Diana, Michelle, whatever your group is made up of, So clarify if it's done independently or done by a group for me. I also noticed through Blackboard, and remember, I'm still learning how Blackboard works on my end as a teacher, but through Blackboard, I'm gonna need every single one of you guys to go ahead and upload a version, so that way in the grading column, you won't have a 0. You may have done the work and then someone in your group uploaded it, but I need everyone to upload it individually. So I may not give comments on every single one of them. I'll just give commentary on one of them, but just make sure that everyone needs to submit it, whether you did it in a group or individually. Okay, so next is the title. Remember, it should not be something like Lab Report 2 or Manduka Project, anything like that. It needs to focus, it needs to communicate what the experiment is about and what your main topic is. So your main topic is the circadian rhythm, and the experiment you're doing is the effects of an organism due to manipulation on the circadian rhythm. So something along those lines should work just fine. And that's 7 points right there, so don't take those. Those are easy points, right? The abstract, remember to use that example to help you, look at it again, see how you would reword it. Your background information on this, though, I will say, is the circadian rhythm. It's not your manduka. So I get that a lot. You know, starts out with a lot of background about what the manduka worms are and all that, but that's not technically your background for this. Remember, you should focus and you should communicate what your experimental topic is, and your experimental topic is the circadian rhythm. If I affect this kind of amount of hours an organism gets of light, you know, how does that affect its activity? How does that affect its growth? So regardless of the organism, that's what you're looking at. And you just happen to use a manduka to represent this. So that goes into your goal, and then your methods is where you want to introduce your model, and your model is the manduka. So your methods is where you give a little bit of background to the manduka. So I know that's a little weird. You're going to talk about the circadian rhythm, the purpose of the study is to do this, and then, you know, furthermore, to conduct this study, we used a manduka sexta hornworm. These manduka sexta hornworms are crucial models because they have the following characteristics. You know, they live in this kind of temperature. They're well, you know, easy to maintain. They're not very expensive. They will go through a similar life cycle, or they go through a life cycle that can be observed, you know, anything like in a matter of 10 days, anything like that. That really goes into your methods, right? I think you guys can see that now. And then a little bit about your results, you know, either the light affected it or the dark affected it, your null and your alternative hypothesis. And remember to use those key words, you know, a null hypothesis of this study is blank, and therefore a alternative hypothesis is blank. Don't assume by not, you know, if you just wrote like light will affect it this way, I can tell that that's your hypothesis, right? But don't assume that your reader will know that that's your hypothesis. You want to be very clear. You want to actually use those keywords, right? Don't assume that you wrote out your goal without actually writing those keywords, the goal of this experiment. So also, more than that, use those little subheaders, if anything, because that will clarify. You know, you can write subheader background, subheader goal, subheader hypothesis. You're more than welcome to do that. Methodology, results, major conclusions. So use those little subheaders inside your abstract and use the example to help you. Okay, so moving into the introduction. So remember, it's background information that relates to your hypothesis. Again, this information, this introduction information, this background should be on the circadian rhythm. You can look at human life cycle, meaning from babies to elderly, and seeing that and how that relates to your experiment. And your experiment is, if we mess with an organism's and we manipulate an organism's circadian rhythm, what is the effect, right? So that becomes the purpose, right? What are the effects of this manipulation? Now, a lot of the rubrics I notice has this, including the question that you're trying to answer. Yeah, that's kind of important, but that should all just kind of be wrapped up in stating your purpose. If you've stated your purpose, you don't really need to state a question because you have stated your purpose, right? So try to avoid doing this. Actually, I'm gonna go ahead and delete it. I think it's there because a lot of times people have trouble thinking of what their purpose is. So if you think of it in a question, you know, what happens if we manipulate this circadian rhythm, then you have actually stated your purpose, right? But say it in a statement and not in a question. So I'm going to go ahead and omit that. You know, state your alternative hypothesis clearly, but in order to describe your alternative, you know, have it right here in the side note. In order to describe your alternative, you have to describe the null hypothesis, right? Your statement right here should sound something along the lines of, you know, a null hypothesis for this experiment is blank, and therefore an alternative hypothesis for this study is blank. And you can fill out those blanks by looking at that manduka handout I went over in the last video. So they should follow somewhere along that handout, or you can use it in your own words as well. Now I put a little arrow here because I noticed that this rubric, again, this rubric's kind of passed down from semester to semester, so I didn't necessarily create it. I've done manipulations to it last semester and now furthermore this semester, but I noticed that it's not necessarily in order. You know, the way it is right now relates the experiment to the real world and then describe the methodology and have 2 citations. It's not the way that I would put it. This seems to like break up right here. I would switch these. So after stating your alternative hypothesis, you know, a null hypothesis to the study is this blank, you know, therefore an alternative hypothesis is this. And, you know, moreover, to test these hypotheses, I'm going to conduct an experiment using the following methods. You know, it flows a little bit more if you describe the methods after your alternative. So go ahead and describe the methods. Remember that here is where you want to introduce Manduka, right? So that quiz that I had you guys do where you talked about what the Manduka was and all that information, all that can go right here in methodology. It is your model of or it's your choice of organism to conduct this study, right? And why it's important as a model and describing its part in the methods, right? So why are the Mandukas such good organisms to look at circadian rhythm? And a little hint to this is that they basically go through their life cycle in a matter of a couple of weeks. So you will see them go from larvae to adult. Basically, they go into the little cocoons at around like day 14. So you'll see them right before then. So you'll see all of that growth in a short amount of time. Whereas if we were to use, let's say monkeys as our a model of experiment, that would take years, right? It takes years for a baby monkey to reach, you know, teenage years, to reach adulthood, you know, and seeing that behavior. So that's not feasible. We could use, you know, fish, so how they go from eggs all the way to adulthood, but fish don't have very similar circadian rhythms to, you know, us mammals, right? So, or us humans. So the Mandukas provide a cheap, you know, monkeys are also very expensive, right? But they provide a cheap, less ethical model to look at the circadian rhythm sorry without waiting years. It's only a matter of days, right? So they develop fast and we're able to observe this development, which is really interesting. So all of those kind of information really can be introduced here. It's introducing your mode of method, right? Your model. You know, and then furthermore, we're going to use the light boxes to look at to manipulate the circadian rhythm or these light cycles. And we are gonna weigh them daily to look at their daily weight. We are going to do some calculations such as the average daily weight or the average growth, describing all that into the introduction. And then putting 2 citations. Clearly, one citation could be for the Manduka, and another citation could be for the circadian rhythm. So that's pretty easy to break down. And now that you flip them, you don't want to end your introduction on the methods, right? We're going to do these calculations to look at daily growth and then compare it to our control period. You know, that's kind of a weird way to end your introduction. So you want to, you know, touch on your major conclusion, and that is, how does this study relate to the real world? How does this study, you know, impact society, impact the scientific community, impact the world? And whenever you see this, you can automatically think, you know, what diseases or disorders are affected by this kind of topic, and will my study help improve these diseases or disorders? So if I, you know, relating this to the real world, if I know that, you know, insomnia is a real thing, and sleep deprivations and, any kind of disorder or disease that correlates with their circadian rhythm. And I know my experiment has has significance, right, to the real world. So ending on that note, researching about that and adding that into your introduction is kind of how you will wrap up that introduction. And then your methodology or your methods and materials we've gone over, you really want to just use that protocol to help you. So step number one in that protocol is to describe the components of your experiment. So again, you need to identify your controls and of your experiments and um, what are your dependent and independent variables, all that good stuff. You can write a paragraph from the start because that's step one. Step two is aseptic technique. Step three is doing this. Step four is doing that. Just use that protocol to help you write out this methodology. Make sure you're not listing or bulleting or numbering anything. This includes when you talk about the materials. If you said the materials for this study is, worms and light boxes, worm food, you're listing them. So that still counts as listing and you'll get points taken off. So instead, what you want to do is write out your methodology. And just anytime you first introduce a material, you want to give details on why you're going to use it and why it's important for this study. And that's how you really talk about, the materials in your methods. Yeah, so if you just use this protocol, that should get a little bit more clear to you. And remember, it's written in past tense, so that is the ed tense, right? It's something that had happened. So you're reporting what had happened. You're not writing a protocol. Protocols are what you will be doing. So it is what happened. And you want to stay in third person. So as a group, we did this. That's not allowed. So you want to stay in third person. So this study uses Manduca sexta hornworms in order as a model for the development of under light conditions, something like that. So that stays under third person. Anytime you struggle with this, anytime you want to say we or our group or a group member or I did this or anything like that, you might want to consider just switching out that word with this study looked at or this research is done or this experiment, right? Anything like that. That's kind of the trick to it. Okay, so continuing on to the results. Remember, in this paragraph, you should really write about what your main findings are. So again, no listing or numbering or anything like that. It shouldn't just be like, okay, on day one, it was the worm one was 3.3 grams. On day two, it was 3.4 grams. On day four, it was this. Like, that's just listing out the stuff. You want to put all that data into a table, right? Which it's already in the table as is. And then refer to it, just like in that last video when I was explaining the tables and the made up numbers, I said, okay, well, we can see that, worm one started to lose some weight. So that's a major finding. So, refer to table one, we can see that worm one started to lose weight at day 4, but then picked it up. This correlates with its slower activity. So those are your major findings for worm 1, and then do some major findings for worm 2 and some major findings for worm 3, and then your overall major findings. So on average, which worm had more effect by light? On average, which worm had less effect on light, or which worm had more effect by the dark hours or less effect by the dark hours, anything like that. That's your main finding, right? Relate it back to your hypothesis. You want to include at least one graph and as many tables as needed. So you can see in the other one, I put 4 tables. You can fill those out. You can add more tables if you'd like. You can add more graphs if you like. This is really showing me that you understood kind of how the different options to report your results, right? We went over several of them throughout the semester, and I went over a couple options in that last video. But yeah, just I'm looking for at least one graph. That, again, I just want to reiterate, that does not mean tables. If you put a table as a graph, those are different. So figure 1 could be that table that I had in that Excel sheet, but that means figure 2 needs to be a bar chart or a line graph or anything like that to show the average growths or whatever. More than that, remember, in your results, you need to show your math. So any kind of calculations that you did, if you did those, in the handout, it has those 2 formulas. Write that down, put it in a table form, and throw that in there. If you're missing that, I will take off points, so make sure you have shown your math. You can throw it into the results and then refer to it inside the paragraph saying, you know, the calculations for daily growth was done by this formula, and the math behind it can be seen in figure 3 or something like that. So you always want to show your math, so that way as a reader I can verify what you did mathematically. Make sure that everything has those descriptive captions, right, that I've talked about a couple times. You can use that example I posted on Blackboard. It's called like lab feedback or something like that. And you can see how I put my results in a text box over the whole thing. I have a title in it, I have a label in it, meaning like I have a figure 1 is the label, and then average growth is the title. And then I have a description caption. So if I did any calculations, you can say like the formula used was this. If you have any major results, clearly the bar graph in column 2 shows the higher growth, anything like that. You want to put into the description caption, regardless if it's repetitive with the paragraph that you put above. You still need to put that information into a descriptive paragraph and a descriptive caption, right? And abbreviations are explained. So like in that Excel sheet that I just went over in the last video where I have A's or VS or anything like that, those are abbreviations. And in that description caption, I would need to say, you know, A stands for active and VS means very slow activity. You know, DLC, D was dark condition, L was light condition, and C was control, where dark was, it was like 18 hours of darkness and 6 hours of light. Light was 18 hours of lightness and 6 hours of dark. And the control was 12 and 12 hours, right? So just explaining those details, these abbreviations need to be explained. And then make sure everything is neat, axes are labeled. This is really important with your graph, right? If you did a histogram, you need to make sure everything is labeled. If you did a bar chart, everything is labeled. If you did a t-test analysis, everything is labeled, anything like that. And that'll be results. And then in the discussion, this is where you would state whether your alternative hypothesis was rejected or supported. And in order to do that, remember, you need to reflect back to your null hypothesis. And yes, a t-test analysis wasn't really necessary for this lab report. Of course, in a real experiment, we would definitely do some kind of analysis that would test statistical significance, like a t-test analysis. And it's totally possible, but I didn't make it a thing for this lab report because really that's to do it twice over, it's a little much, right? So instead, you can decide whether you accept or reject your null hypothesis and therefore accept or reject your alternative hypothesis by looking at those averages and comparing it to the control. So if the dark is higher than the control, or if it's lower than the control, then you can reflect back on your alternative and decide, you know, did you see what you had predicted, right? You know, explain this support and reasoning for the decision. That's just going to go back and, like, you know, based on the average calculations that I did, You can see that I predicted correctly, that therefore my alternative hypothesis should be accepted. Anything like that. Explain your major results meanings. This should be your one-liner. So this should be, darkness had less growth, or light condition had more growth, or the control condition had more or less growth, which should not be seen. Anything of that. What is your major one line result? Which condition gave you a higher growth? Or it just depends on your hypothesis. If you decided to look at darkness as a hypothesis, which condition gave you less growth, right? A decrease in growth. So let's see here. How do your results fit into the real world? So this is touching back on, your discussion. Your discussion is also like your conclusion. You want to wrap up and say, this study has so much importance in the real world because of certain conditions that can happen to humans if you manipulate their circadian rhythm or anything like that. Again, I would probably put this kind of stuff elsewhere. I'd probably put this down at the end because it's a nice way to end your paragraph. So I can put in another arrow and put this like towards the bottom. Again, this isn't necessarily in order. This isn't how you would, list out your discussion. For the most part, it is. You do want to start off with, what happened to your hypothesis based on these results, what do your results mean, and explain this, but you can kind of, you can explain your results first, you can then state your hypothesis, then give reasoning. So you can kind of switch around these points. Don't think they're in order. Compare your results to other research. So you want to make sure there's some other citations out there, maybe that used a different model, or maybe that used, slightly different light conditions. Maybe they didn't use a light box. Maybe they used something else to control those light conditions. You want to look at that and see did they have similar results or did they have different results. You really want to look for a paper that has similar results, right? Because that backs up what you saw in labs. So that gives more evidence to your results. That gives your discussion or conclusion more power if you have something that kind of correlates to what you did in lab, right? Mention your weaknesses or your biasness. Again, don't undermine your methods or your main goals. So if you're mentioning the weaknesses and you're saying, oh, we should have used fish instead of the Manduka hornworms, then you're undermining your methods. You're saying that this whole experiment, regardless of the results you had, was a waste because you really shouldn't have used the Manduka. So you don't want to undermine your, and that's not necessarily true because you had good results. You should have good results come from this. So you don't want to necessarily, any kind of mention about a different step into the methods is not necessarily the way you want to go. Or you don't want to switch up your main goal. So maybe instead of light conditions for circadian rhythm, we should have looked at I don't know. We should have looked, instead of light conditions, we should have looked at their eyes or something like that, and their eye movement or something like that. That's a different study altogether. You want to stick with your main goal. You wanted to look exactly at growth conditions on the different light conditions, right? The different light variables. So don't change up anything like that. When you mention weaknesses and biasness, you don't want to undermine your methods or your main goal. What you could state is, maybe this wasn't a good enough range, right? 18 hours of darkness and 6 hours of lightness. Maybe, furthermore, regardless that we got good results, the next time we do this experiment, I would want to look at what happens with 20 hours of darkness and only 4 hours of light, or 20 hours of lightness and only 4 hours of dark. Like, so those extremes. So, you know, you don't undermine your methods that much by saying that, because you're saying, you know, we got good results by the way we did it, but I would just want to add on more. So I would still add that 18 hours of darkness and 6 hours of lightness in the next round. If I were to have repeat this, I would still have those steps. I would still have the same methodology, but I would add in more light conditions or more dark conditions, just to see at the extremes what happens. And then suggestions for future research. So that's not really a suggestion for future research, you know, just changing up a little bit of your protocol or your purpose isn't a suggestion for future research. For future research, you want to look at your main findings. So if this right here, if this one line was, you know, sorry, if this one line was, you know, more light hours led to an increase of activity and an increase of growth in the Mandukas organisms, then what can you do next? You know, maybe you want to see, maybe you want to dissect that worm to see, and gather its brain to see different hormones involved in its brain, maybe different structures developed in its brain to see if any changes happened there, because your result was an effect in that one worm. So now let's do something to see what happened in that worm, to really clarify on what happened. So what's the next step? So that's how you need to see what the suggestion on future research is. It's what's the next step? Now that you know what you know, now that you know what you have discovered, right? You discovered what condition was best, right? What's the next step? You know, maybe the worm in the dark had way less activity. Maybe it had, you know, shrinkage and growth. It lost weight. It was very slow activity. At the end of this, the next step would probably be to dissect that worm and to further look into the reasonings behind this. And that'll take a little bit of research to suggest what to do next. So if you do put the lines on like, oh, I need to dissect him, you need to give more than what, okay, well, what are you going to look at this dissection? You know, what parts of the brains might you look at if you're looking at circadian rhythm? What parts of, you know, which cells are important for sleep? What hormones are important for sleep and activities and anything like that? So this will take a little bit of Google searching, but it shouldn't take much. And of course, you need a one in-text citation, so that could be done. It's at least one, right? It can be done for this future research, or it can definitely be done for comparing your results to other research. You need to cite the other research that was done, right? So there's one citation there. If you have any other ones, because you looked up like different methods for future research or dissections or whatnot, you can cite that as well here. And speaking of citing, remember that your reference page goes on the next page, on a page on its own. It'll have at least 3, because you have 1 in your discussion and 2 inside your introduction, but at least 3. A lot of you guys gave me more than 3, which was great. Make sure it's an APA format. Make sure that it has your in-text citation. So anytime a reference sheet is provided, you should have those in-text citations as well. So when I'm reading your paper and I see, you know, according to this paper, yada, 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 and in those parentheses, I see that you put an in-text citation, I should be able to grab that in-text citation and refer back to your reference page, right? That's why it's called a reference page. I'll refer to it for the full citation, and then I'll be able to click on it and see where you got your information from, right? So these in-text citations are really important, so make sure you do them properly. And then make sure it's in alphabetical order and that your lab manual can be listed as one. So if you have, you know, the information on the Mandukas, that could probably come from your lab manuals. So you might want to use that one as one of your references. All right, and then overall, I'm looking again for Very few grammatical errors, especially now that this can be done as a group. There should be very little grammatical errors because you all should check on each other's sentences and any issues, right? You want to make sure that everything's in the proper tenses, no IUS. We kind of talked about that already. Everything is neat and organized. Everything has those headers. Everything's double spaced. Remember that headers go to the left and they're typically bolded unless they have a sheet on its own. So the only headers that go centered in the middle, bolded, are your abstract, right? That header will be centered in the middle and bolded, and the reference page center in the middle and bolded. Everything else is aligned to the left. Everything is double spaced, and everything's neat and organized, right? Those results are in its own text boxes. Everything has been labeled in those results. Everything has the descriptive captions. Everything's referred to in those description captions, anything like that. We already talked about the extra credit, and you have a final score out of 100, possibly out of 110, right? So more than that, I guess my last little discussion here before I wrap up this video is that if you do it in a group, and this kind of really relates to here, make sure it sounds like you guys got together and worked in it as a group. And what I mean by that is, like, typically group reports are usually broken down, right? We learned this in, like, I don't know, high school or something. Like, okay, you do the introduction, I'll do the methods, and you do the conclusion, and you do the results. And that's fine. But then what you get is a paper that has certain style to it or a certain voice to it in the intro, and then a different style in the results. And then you also get a lot of repetitiveness because you don't even tend to look at what the first person wrote in the introduction. So you tend to repeat yourself a lot in the results, or repeat yourself a lot in the discussion. And it's also a little bit harder to do it that way because the results guy might do a certain thing, and then the discussion guy might not understand what the results guy was doing. So you really just need to work as a team. I'm not saying that you can't do it that way, that's fine, but then I would make sure that one or two people are, if you break it down, like, okay, you do the introduction, you do the methods, you do the results or whatever, then you need to make sure like one or two people are also in charge of reading it from top to bottom, or everyone can be in charge of this, but everyone needs to read it from top to bottom and make sure it sounds like it's one report, like as if you guys got together in the library, sat down at a table, and you guys wrote out the report together. So it needs to sound that way. So the other way you can do this is just by having a Zoom meeting or a FaceTime or a Skype meeting, anything like that, and just working at it as a group. So it sounds like one voice, or you can do it independently and it'll sound like one voice. So that is something to be definitely aware of as it's a group project. And that's it. That's all I have to say for the SILAB report 2. If you have any questions, go ahead and email me. In the next video, I'm going to go over basically anything left for the semester. So be sure to watch it as well. All right, bye.